Thank you. So, well, it's my pleasure to be here, especially because I didn't present things for EBD in a while. So it's very nice to present it in this place. And I want to try to do an overview of a few things that we are doing in our lab right now. So you get a flavor of what kind of things we are doing, all of them related to bees and pollinators in general. So before that, I would like to start on why we are here. And one of the main things that motivates us to research is having at least a voice or an impact on current problems. You all know we are changing things very rapidly. So we are a lot of people. We also, it's not just that we are a lot of people, but we also have a lot of activities that are changing the environment, including increase on CO2 levels and temperature increases. And most importantly, we're also really good at destroying things. So we go to very nice forests, we clear cut them, put agricultural systems, and all these things have an impact on biodiversity. So the overarching framework of our lab is how these global change effects are affecting community composition. And for that, we need to understand both how ecology works, how ecological communities work, but also have a view on conservation and try to manage all these things. So I'll try to talk a bit about conservation, which I think it's an important topic to cover related to that. Also, a few examples of how do we use ecological theory to answer some questions, especially regarding network theory, which we like a lot to use as a framework to understand communities. And then I would also like to talk a bit about applied ecology, because I think it's also an important part of our research. And as you will see, I hope, these are not different compartments, but I would like at least to use theory to inform conservation and applied things, and they talk to each other. What I'm not going to talk about today, about bee brains. But if you want to hear about bee cognitive ecology, Miguel Ángel Collado will be defending his PhD thesis the 25th of October in this same room. So please join us, and you will learn a lot about how bee brains work. Okay, so let's start. First thing, if we need to conserve something, is we need to know what's there. What are the trends on the populations? And that is not always easy. For bees, probably you already know, but are a very diverse group, more than 20,000 species worldwide. Just in Spain, around 1,000 species. So it's, for context, kind of twice the number of species of birds. And they have very different life histories, small, big, parasites, specialists, social species, solitary species. So a huge diversity. And when all the pollinator crises start taking out this idea of maybe pollinators are declining, the National Academy of Science put a commission together, like, okay, can you inform us about this? And this was the main conclusion. Basically, it said that for most pollinator species, there is no long-term data. We don't even know the basic taxonomy or ecology. And basically, who knows what's happening? That's 2008. I think we've advanced a bit since then, but the main take-home message is still the same. It's very hard to know what are the bee population trends. So this is a map of bee diversity. The reddish, the more diversity you can see, Mediterranean areas in South Africa, California, Mexico are highly like are hotspots of biodiversity. And this is the countries for which we know at least something about how this diversity is changing in the last years. So it's very small, but Britain, Netherlands, US, and a few places on Brazil. The rest of the world, we know more or less nothing. So how do we do that? Can we go back to the past to know how bee populations were back then and compare them to how they are looking now? I didn't find a funding agency yet that allowed me to buy a DeLorean. So our approach is a bit more pedestrian, but if you want to know things about history, you go to museums. And this collaboration started a while ago in the American Natural History Museum, where I collaborated with John Asher and Rachel Winfrey to database on bee species. The museum is amazing. I have these big dinosaurs, but you can walk all through the museum. There's not a single bee displayed at all. But if you go to the basement, like 3 floors down the earth, you can find John Asher, who has a incredible sample of bees from around the world. Why museum specimens are so useful? Because collectors are crazy people, crazy in a good way. So they're very systematic. So they really like to have things in order and tidy them. So they know when they collected the species, who did that, where. And from this information, which doesn't look like that many, we can understand things about phenological records, changes in abundance, or sometimes they even record the interactions. So this is a surprisingly nice data resource. So this is the only old stuff I'm going to present for context, but what did we find when we analyzed these historical records in the United States? So in general, there is a trend to decrease species richness. This is the number of species that we found in different time periods. And just for your knowledge, this is rarified richness, so we try to control for as much sampling biases as we could. And I'm not going to get into details on the analysis. Some other groups, like the bumblebees, have stronger declines. And that's something we knew from other sources, and it's very nice that the museum records also show that. But something also very interesting is that some other species are increasing, for example, invasive or exotic species. In the 1900s, there was barely known exotic bee species in the United States, while nowadays there is more than, I think it's already 18 species or something like that. So this is the kind of trends we can get. We can also try to understand specific species, like it's not everything is about species richness, but how some species are doing. So we can calculate the rate of change for each individual species that we measure. So we get up to 187 species with enough data. And we can see there is some phylogenetical signal. So some genus, like these Ceratinas or Nomadas, are doing pretty good. In fact, they are increasing its relative abundance. Those species are also the ones that can live in agricultural areas or in urban environments. Some other species, Coleoxys, megachilids, bumblebees, are having consistently negative responses. So those are the losers of these changes we're observing. And that's something very interesting we can get. So starting from there, when I moved here to EBD, we were thinking, okay, that's great. Can we do something similar for Spain? And we don't have such good collections, especially that are database, because there exist some collections, but they need a revision and taxonomical revision and databasing, which it's a lot of work. But we also have very good naturalists and scientists in the past. So working with Enrique Asensio and Oscar Aguado, Enrique Asensio was a pioneer in bee taxonomy and ecology in the 70s, and he systematically recorded the bee community in 6 localities nearby around Valladolid. So with Oscar Aguado, we went back with the same methods, same areas to test how things are going right now. And the answer is not too encouraging. So species richness overall has decreased. It's nothing too surprising because a lot of things have changed in that time period. But I want to highlight 2 things. And are these 2 dots here. These 2 communities will recover barely all the species that Enrique was monitoring were still present nowadays. So while the general trend is clear, for me, this is very illustrative, and it's hard to make statistics with 6 data points, as you may know, but we can still get a lot of information from that. Those 2 places are places in the Páramo where very little changes have occurred in the last years. In fact, if anything, agricultural pressure has decreased because there was the less productive lands. These other places were sites that get an increased urbanization, habitat loss, and increased agricultural pressure. So while the overall picture may seem worrisome, I want also to stress that I think most of the bee species, at least in Spain, are still there. And this is great news for me. Like, very little extinctions has been reported so far. This means that if we start thinking on conservation, we can recover these populations. And thinking about these pockets of natural areas that remain and that can sustain nice populations. The example I'm using lately is that to conserve the lynx, it's very complex because you need to conserve huge landscapes. But to conserve the bees, it's way easier in a way because a lot of the populations can thrive in places around like half hectare or a quarter of an hectare would be enough habitat for getting healthy populations. So there is a lot of room to improvement on insect conservation. Then I got a bit ambitious and I said, okay, we know a bit about United States, a bit about Spain. What do we know of the rest of the world? Can we do something with the tools we have? So I went to GBIF, probably you all know, have a lot of records from very different species and time periods. And I retrieved all the data reporting bee occurrence for all countries. This is all 125 countries in the world. And this is just, you can compare the records, existing records prior to 1980s and recent records. Very crude first analysis, like we have enough data. For those group of countries there is no data, neither historical nor recent. Some other species have, countries have good recent data, but not historical one, so it's hard to compare. But there is a lot of countries, I think around 22, which already have digitalized a lot of occurrence for recent time periods and for all time periods. And again, this is a very crude analysis. It's a very first order thing, a proof of concept to see, can we use this data at larger scales? But one of these thoughts is a different country, and consistently, most of them, except but 5, we found moderate decreases in species richness once controlling for sampling effort. Again, you can take that as good news or bad news. Bad news, we have a consistent decline. Good news, this decline is very is mild, so probably those are not real extinctions. Those are just species which populations go beyond detectable levels in this kind of very coarse data. But this allows us to feel a bit more this map and try to see a bit clear, like when we read in the news, there is a worldwide pollinator crisis, and my mother calls me like, are we doomed? Are we gonna die? And I can say a bit more about, no, this is just about a few places in the United States and Europe for which we have data. Now at least we can see that this is quite widespread in places like Japan, India, South Africa, and it's more represented. Australia, New Zealand have data. And also the positive trends tend to be in the north, which is consistent with a global warming scenario where these places are getting better conditions for species that like the heat. Okay, so this is part of the things we are interested, especially trying to get this information to make an impact on conservation. And I would like to show you a few things that we started here in Doñana, and some of them are a bit preliminary, but I thought it was a really good audience to show what we've been doing. And it's about interaction networks. So I don't know what you see here, but this is not a typewriter. It has all the components to create a typewriter, but they are disassembled. And they are hence, therefore not functional. The same happens with ecological communities. So those are a bunch of bee species, but they are not a community. A community requires to know which are they feeding on, which are the interactions, what other pathogens or parasites or predators they have, and also how they interact among them. So we need to build these networks to understand community ecology. And this is what we started to do. This is what we started to do in 2015, along with my friend Curro Molina, who you can see catching bees down there. And we selected 16 forest islands in the Guadalquivir Valley. Some of them are tiny, refuges of tiny patches of forest that remain there in a highly agricultural area. Some of them are huge areas. This is Aznalcázar, this is Hinojos. So very nice, well-conserved areas. So we can compare how the communities look like among them. And the idea is that we know barely nothing about temporal dynamics. We just, as ecologists, our PhD theses are short and funding schemes are short. We go, we sample, and we try to make inference from there. So here we can, we aim to get some temporal information. So we already have 5 years of data and counting. What do we do there? Basically, we spend a lot of time watching the flowers and measuring which bees visit each flower, so we can build a network of interactions. And with that kind of information, we can do a lot of things. In fact, we still didn't do much because we were waiting to get a longer period, but now with 5 years, I think we can start doing stuff. So I will present 3 types of ecological relevant questions we can answer with this kind of data. The first one is relating network structure to community stability. So there is a lot of emphasis on that how this network is assembled should tell us something about how the community is able to resist some impacts. However, most of this work is theoretical, and there is very few data to show that. So today I'm not going to show too much data, but at least there are some preliminary analysis, so it will serve also to give you a flavor of the system. So this is the first year, 2015, and this is the abundances of plants and pollinators. So this is Southern Europe. It's warm climate. Around 10th of February, things start flowering, and the activity of both plants and pollinators start. It's a bit bimodal, and so far so good. That was my first year of data, and we thought, okay, we understand more or less the system. This is the peak of phenology. Great. 2016, I was driving back from home at Christmas, and I started seeing, like, this plant is flowering there, this plant is flowering right there, and I called Curro and said, grab your stuff, we're going to the field, like, 8th of January, like things are on fire. this was a really dry and warm year, so things start almost a month earlier. And as you can see, pollinators were a bit faster to respond, but both of them respond quite simultaneously. So also the length of the season was really long for us. For the bees, probably was okay. And I'm going to show 2017 again a bit earlier than 2015, but a more normal distribution with some mismatch here, but probably not too relevant. So with a hard analysis, first thing we can say is that synchrony is not perfect, but it's not lost with extreme weather events so far. That's really interesting. I'm looking forward to add 2 more years of data and do a more formal test. But there is a lot of hype about synchrony will get completely mismatched between interacting species, and this will cause problems. But so far, this is not what we are seeing, at least up to now. We can also look at the pollinator populations. Again, this is just 3 years of data I'm going to show today. But 2015, we get around this level of pollinator density. 2016, as you remember, the year very dry, very few flowers, very warm conditions. We found a significant decrease in the population density, which was well, it's relevant for conservation purposes also. 2017, it recovers a bit, but still we see kind of a time lag response on the next generation. Again, soft data, 3 years. It's a lot of work, but still 3 years. So let's see how this looks like with 2 or 3 more years of data. And this is very similar. It's visitation rates. So we have 2 independent kinds of data, pollinator densities in transects and visitation rates in focal observations, just to reinforce that the pattern is the same. Flowers are getting less visits in bad years. And so the second thing we can infer so far is that pollinators and plant visitation rates suffer from these heat waves or extreme weather events, even if the phenological synchrony remains. And second, and getting a bit more on the networks, we have some years with huge plant resources. This is a very sketch of a network with different species of plants and different species of pollinators and how they are connected. So what happens when one species was very abundant in one year and the other species was, and the same species is less abundant in another year? Do we find some rewiring of interactions, or do pollinators stick to the same? And we can see these abundances of fluctuation in abundances for different groups. So the first group is a Flavipanurgus. It's a highly specialist, only visits Thistus crispus, highly specialized. The abundance were quite homogeneous. So Thistus crispus is also a very abundant species in our system. So not surprising. And of course, there is a full overlap in their interactions because it only visits Thistus crispus in the 3 consecutive years. What about a highly generalist species like Bombus terrestris? We see also the populations are quite stable, but in this case, there is a turnover of interactions between these 2 years of around 10% and similar in these other 2 years. That means that depending on which floral availability is out there, they can switch preference. And that may allow them to maintain population levels high. Finally, an intermediate species, generalist, but not as much as Bombus terrestris. That one, it really got down in 2016, but the turnover of interactions was huge. So in some years, they were visiting some plants, and in some other years, they were switching their diet. Again, not much answer so far. So interactions are plastic, which is great, but again, it's hard to predict the consequences, because even with the plastic rewiring, we can see a decrease in the population size. What else we can do with this data? with this data, we were interested also in knowing the ecosystem functioning. So in our context, mainly ecosystem functioning means plant reproduction. And there is a lot of papers and theory about how biological diversity increase ecosystem functioning. And Bradley Cardinale and all these people has done a lot of experiments on that. But how the species interacts, the structure of the network is also predicted to influence the functioning. So this is what we do. We got data from one of the years. We get the pollinators and the plants and the links. And we also have plant reproduction. Here it's just an example of a seed set, random seed set. So we can relate the structure of this with the plant, the function received by the plants. And we can do that at an individual level. So these nodes, where you are within the network, if you are more connected or less connected, or we can do that at the network level, at the community level, if the full structure of the community. And I'm going to show the results directly because they were quite interesting for us. In fact, where you are within a community is very relevant for your fruit set. So we get, I think it's 21 species or 19 species of plants where we get the fruit set. We know which position in the network they occupy, and we measure the centrality. Centrality is basically a measure of how well connected you are with the rest of the community. So species that are really well connected get higher fruit set. And of course, the variability is huge here. But there is a pattern on how I am connected with the rest matters to me. And this is in addition to simple metrics like species, how many partners do I have, or things like that. At the network level, of course, here we just have the 16 data points of each community. We also see that the overall fruit set, the mean reproduction of all plant species, is correlated with the pollinator niche complementarity. So communities where there is more overlap and all pollinators are visiting all the plants are more messy and get lower fruit set overall. The mechanism is hard to predict, but maybe there is more heterospecific pollen deposition, there is less consistency. And communities where there is more niche complementarity, so some pollinators visit some plants and some other pollinators visit some other plants, those plants get higher reproduction. So that was very interesting for us, and as far as I know, it's one of the first examples where we empirically show something that's it's been demonstrated theoretically. The third example I want to put using this same data set is about to measure competition. Measuring competition is hard if you try with any system. So networks also give us an opportunity to get the indirect signature of this competition, when we can observe if what we observe, it matches the predictions. As I said, pollinators compete among each other, but usually this is ignored in the literature because it's so hard to measure. But there is one scenario where this is very relevant, or easier to test, is when you have one major pollinator which can exert a huge competition on the rest, which means honeybees. And this again touches on conservation because we need to understand these processes to advise how we manage the natural systems. So with Ainhoa Magrach, we compare areas with very low honeybee densities, areas with high honeybee densities. And I'm going to summarize this with this sketch. But this is how the average network looks like. I'm going to walk you through. So the links, each of these lines, are more or less evenly distributed. So each species have a similar amount of link intensity with other species. Of course, there are some links that are more important than others, but not huge differences. Then we have a high number of interactions per species. So each bee species can visit 3, 4, 5 plant species. And third, there is low niche segregation. So as I said, a lot of these pollinators are generalists, so they visit a bit of everything. What happens, and that gives to low apparent competition. What happens when there is a lot of honeybees? With a lot of honeybees, they interact a lot with some plants, but only with a few of them, the preferred one. And this creates a very high and even distribution where there are a few links that dominate everything and the others are very rare. Moreover, when the niche of each pollinator is reduced. So if I can visit 4 flowers, now I tend to visit only 2. And these 2 flowers that I am still visiting are not random. They are the ones that the honeybees is using. So I try to avoid the resources that my competitor is using. We had some back and forth with the reviewers if we were showing competition or not. They were saying demonstrating competition needs to be experimental and really hard to do, which I agree because it's hard. But I think we can show all the signatures that we expect from a strong competitive effect. Which is another way we use networks to merge between ecological theory and conservation. Okay, that's the last example I would like to use, and I think I'm more or less on time. Yeah. And for me, it's also very important, as I said, to make things that are relevant, and with a large network of co-authors, we are working on understanding pollination in crop systems. Some of those works are more descriptive, like just putting the relevance of pollinators for crop production, which until recently was a thing of something that was secondary in comparison with fertilizers, pesticides, or water, which now I think with our work we put them in the first front, like pollinators is one of the key regulators of crop production. But in these systems, there are also interesting questions, and questions that are highly linked to ecology, and we need the ecological tools to answer. So, as I said, there is normal relation between biological diversity and ecosystem functioning, and most of these studies are experimental. This is great because we need to start somewhere, so people just were planting a bunch of different plots with different number of species. Some plots have 1, 2 species, some other plots have 20 or 16, 20 species maximum. And they look at the functioning, which usually is biomass in the plant world, and they see that the more biodiversity, the more species, the more function you get. Which is good, because it puts in value the importance of having a high numbers of biodiversity. But this is one of the most important of these experiments. This is Cedar Creek, run by David Tillman. And each of these quadrats is one of the settings that I said. And this is the scale at what they work. But what happens if we want to predict ecosystem functioning at the full scale of the experiment? How many species do we need to maximize functioning? This is still the same experiment, and this is the landscape around. What if we want to understand which biodiversity do we need in the landscape? Are those experiments scaling up well? Because those experiments, those is a figure from a Nature paper from Bradley Cardinale, has no access, but usually they asymptote quite low. At 5, 6, 7 species, they got maximum production. This makes sense at the ecosystem level, or do we need more there? So how many species do we really need for provisioning these ecosystem services, like crop production? This is both an interesting question from the applied perspective and from the basic perspective. So that's what people were thinking a couple of years back. Like, now we need just a few. With a few species, we are fine. Why? This is how the experiments look like. Each species would have the same abundance. They put them in experiment, and they look at the importance of biodiversity. But you know that in nature, species don't have the same abundance. That's how most species look like in nature. Log normal distribution, right, David? So these species will be contributing a lot, while these species will be contributing almost nothing. So we need less species than we think. That's one option. But other people were thinking that we need more species than we think. Like, biodiversity is even more important than these experiments are showing. Why? Because this is a classical experimental setting. We have 6 species here, 1 less here, and a few less here. But the species identity was the same. It's nested in a way. So you just remove a species, but you get the same species. Again, as ecologists, we know that when you go to the field, this is not what it happens. When you go to the field, you have a lot of high species diversity here, intermediate diversity here, but there is also a replacement. And here you have only 3 species, but 2 of them are different. So if you want to conserve, to know which biodiversity is needed at this scale, the scale of the gamma diversity, these species will be important, even if it's represented only in one place. So if we take into account beta diversity, we need more species. So there is this two opposing forces showing that if turnover is very important, biodiversity will be more important than we think. But if dominance is what really drives the functioning, and the dominant species are quite common everywhere, then we will need less than expected. So we tried to figure out which of these 2 aspects were more important, either turnover or dominance. And this we started a while ago. So we compiled good data in 3 crops: watermelon, blueberry, and cranberry. So we could, in real landscapes, understand how this works. And this is done the data comes from the United States. And you can see there are several places around which we can combine. So 50 sites each, and we can measure the function in a very elegant way because we have the visitation rate. We know how many visits each species is performing. But we also know the pollinator efficiency, so how many pollen grains it deposits on the flower. Counting these pollen grains it's super fun. But something you need to do if you want to understand actual functioning. So for each species, we know the contribution to the function, which is pollen delivery. We know how many visits and how efficient they are. So we can get the efficiency per species and the complete efficiency of the system. And the graphs that I've been showing you so far is how species richness increase function. So we want to change a bit the question. So instead of looking at this, we will say, okay, which is the species richness needed in this community to maximize function? And we plot it here. So across different communities, how many species do we need to fulfill function in all of them? And that's the key process. We don't want an average here, but maybe you need 3 species to pollinate the first crop, the first site, and 4 species to pollinate the second site. But these 3 plus 1. And we can see how this asymptotes. And I play again this figure in a bit more, so we can get how the number of species increase with the number of communities considered, so as the scale increases. And this is the observed data. And to compare that, so this would be turnover. So this observed data would be turnover. The more steep this is, the more new species you need as you get to another site. So when you visit the next site, they will have new species, so you will need to add these species to the list. So these blue lines tell us about how important is turnover, which is more or less easy. How do we take into account the dominance? So we get a null model for that. So as I said, we know how many visits each species it does and how many pollen deposition it does. So we can create a null model saying, okay, if I have 20 species, let's say all the visitations are spread equally and the efficiency is maintained in this case. So there is no dominance patterns. We attribute to each species the same abundance. It's like making an experiment in a machine, but it allows you to answer how that would looks like in an experimental setting with no dominance. So with no dominance, you need more species, and this is the effect of dominance. I don't know if that was clear, but the take-home message is we're now to compare these kind of figures, and we want to check which one is bigger, the blue or the green line. And this is also for watermelon. We go from 1 to 15 sites in watermelon. And we can see as we consider more sites, we need more species. So the turnover effect is huge. And the dominance effect, it exists, but it's moderate. So it's way more important turnover than the dominance effect. And this is the same that we found for cranberry, even more drastic, the turnover effect, and the same that we found for blueberry. We can even go further and say, okay, what if I want to pollinate all 3 crops? What would be more important? For pollinate all 3 crops at the same time, turnover is clearly overwhelming the effect of dominance. So the answer is that we need way more species that we think, basically because turnover is so prevalent, at least in our system. And this is only taking into account 15 sites and 3 crops. And the number of species needed increases by 3. So what if you want to pollinate all North America for all the crops that depend on pollinators? Probably the number of species that you require in each place and crop would be huge. So this is more or less what I wanted to talk today about. And of course, this is thanks to my network of collaborators, the lab, which did a lot of this work, and funding agencies. And thank you very much.